The question that we wanted to ask is how can we simulate whole brain activity across time? Assuming that the brain is a deterministic system, it makes sense that we should be able to model the activity of a particular brain region at any point in time as a composite of activity from other brain regions at a previous time point. This is the theoretical approach that we used. First, we looked at functional connectivity data. However, functional connectivity data tells us the correlation between brain regions, and that's not exactly the information that we're looking for. What we want to know is the contribution data. We want to understand how much the signal from one brain region contributes to exciting or inhibiting other regions of the brain. To obtain this information, we performed a Cholesky decomposition. It's a matrix decomposition that re-expresses the functional connectivity as the weighted contributions of regions to each other. The key point here is that we're moving from the realm of correlations and into the realm of contributions between brain regions. The result is what we call a weighted connectivity matrix. When you compare the weighted connectivity matrix obtained from the Cholesky decomposition to the full correlation from traditional functional connectivity analysis, you can see that there's greater specificity in the contributions of brain regions to each other. Further, when you compare the Cholesky decomposition results to partial correlation analysis, you can see that the long-range connections in the brain are better preserved, and also that the whole brain composition is more clean through the Cholesky approach. We then used our weighted connectivity matrix in a two-part simulation. First, we represent the activity level of each brain region as the weighted sum of all other regions from a previous time point. Second, we normalize the activity of the brain in order to prevent unrealistically high or low absolute activity levels. We ran the simulation on the functional Connectome 1000 dataset, and after 40 steps in the simulation, this is what we observed. A clear convergence to the default mode network. On the x-axis of the graph on the left, you have the number of steps in the simulation, and on the y-axis, you have the change in brain activity across each step. We need to know, though, whether the convergence to the default mode network is an artifact of averaging all of the brain scans or whether it's reproducible across datasets. To assess this, we looked at the Beijing dataset and compared it to the Cambridge dataset. And in both cases, we saw clear, robust convergence to the default mode network. On closer inspection, not all of the trials converged immediately. Many of them showed an intermediate, transiently stable configuration. And so the logical question is, well, what do these intermediate states look like? And as it turns out, they look very similar to well-defined intrinsic connectivity networks, such as the visual network, the salience detection network, the attention control networks, the sensory motor network, and major hubs of the default mode network. To take this a step further, we asked whether this is reproducible in the single subject, because this is where we really start to get clinical relevance. It turns out that when we compare a single subject who participated in cartoon watching for several hours and subsequently participated in a wakeful rest for several hours inside of the scanner, that there was a clear distinction in the convergence patterns between the two tasks. With the cartoon watching task, there was a high reproducibility for all sessions in which the subject was engaged in this task. At the same time, when the subject participated in resting state acquisitions, there was a clear similarity in the convergence patterns of the brain activity from these tasks. When we correlated the cartoon scans with the resting scans, it was very obvious that each particular task was clustering with a high correlation to other scans obtained during the same task. This raises great questions about whether we might be able to ultimately apply this for some type of diagnostic criteria, or whether this can give us information at the single subject level, which is, again, where it becomes really relevant clinically. For more information about the methods that we used in this paper, please refer to the NeuroImage publication entitled Dynamical Stability of Intrinsic Connectivity Networks.